So we're really pleased we have Ravinder Nagpal, who's a new assistant professor of nutrition, food, and exercise sciences in the College of Health and Human Sciences. Hopefully I'm going to remember health and human sciences. And his research focuses on primarily understanding host-associated microbiome. What's that? He'll talk about it shortly if you're not familiar with the concept, and the dynamics of diet-gut-brain interface at the extremes of aging and an aging-associated metabolic and cognitive health. And his lab aims to discover what he calls novel signatures of gut dysbiosis in host metabolic diseases, including things like obesity, type 2 diabetes, and disorders including stress, anxiety, and depression. So the aim is to explore how the microbiome might modulate specific, with the aid of specific nutritional interventions, including dietary elements, micronutrients, probiotics, prebiotics, how those could all ameliorate any pathology of those particular, associated with those particular aging-related disorders. And I mentioned before we went on, I just undergone a large dose of antibiotics to try to kill a cough I've had for about a month. And so he was giving me some sound advice on how to repopulate my my gut with the right sorts of microbiota. So I'm going to turn it over to Ravi. Thanks again for joining us, and go ahead. Please share your screen.
Thank you, Dr. Chanchan, and good morning, everyone. Thank you for this chance. You know, as you said, I'm pretty new here. I don't have a whole lot or a bunch of data from FSU, but so today I'm going to present some of the data which are related to the association between diet and microbiome, how different diets and dietary factors influence our microbiome. And most of these data have been generated during my postdoctoral studies at Wake Forest School of Medicine. But I'm going to present only those data today, some of which are going to be carried over by me during my ongoing research at FSU. So let me quickly share my screen. So I hope everyone can see my slides. So
yeah, we can see them. Go ahead.
So like past couple of decades have clearly shown that actually our bodies, like we as a physiological system, we are more microbial than human, considering that at any point of time, like our bodies contains like 10 to 100 trillion of microbes on or inside our body. And these microbes, like we have seen, that they outdo our own eukaryotic cells, not only in terms of number of cells, but also in the genome size. So this community of microbes associated with our human body, we call it human microbiome. And we have seen that 95% of these bugs associated with us, they reside in our gastrointestinal tract. We call them gut microbiome. And immense research has clearly shown recently that bugs residing in our gut, the gut biota, has been associated with almost every disease associated not only with gut, but also outside the gut. For example, neurodegenerative problems, cardiovascular problems, digestive diseases, cancers, muscular issues, as well as lung problems. So clearly showing that, yes, gut microbiome has a direct or indirect connection with a lot of human diseases. My research is currently focused mainly on, how early life microbiome is developed. What is our microbiome when we are, when we come to this world, when we are born, and how early life factors like mode of birth and mode of feeding, they shape our microbiome during first 1 to 3 years of life. In addition, I'm focusing on my research, how our microbiome is altered or changed when we become very old, like 65, 70 plus, how people who are aging, who have senescence going on, how the microbiome is involved in that, and whether and how we can ameliorate those aging-associated biome signatures to improve aging, to promote healthy aging and longevity. So in addition, I'm trying to do some studies on understanding how different prebiotics and probiotics, how they regulate the microbiome and metabolic health in mouse models. And in addition, one component of my research is how diet-induced microbiome changes affect the neurocognitive health in different mouse models. So this is a very crowded slide that showing, illustrating that how our microbiome is actually altered during different stages of lifespan. My research has clearly shown that our microbiome is very different when we are born or during gestation staging and during first 3 months to 3 years of life. And this tend to be very different compared to microbiome in children and adults. And then in old people, the microbiome again changes drastically, especially after 60 to 65 years of age. And here is a big list of factors that influence the microbiome at different stages of life, and there are unique factors at each or every stage. But there is one factor which is common at different stages of life, which always regulate the microbiome, and that factor is diet. And that's where like my today's talk is going to be focused on, like how diet influences the microbiome. So as I said, the diet is one of the primary and profound factors tightly regulating and shaping the microbiome. Whatever we eat, whatever goes in our gut, just changes the microbiome. And sometimes these microbiome changes are very quick. They can be seen even in a day or two. Other than diet, there are pharmaceutical compounds like drugs, antibiotics, steroids, and many other things, melatonin. They have been shown to change the microbiome. Our geographical location also go on what kind of microbiome we have. We have seen that people from India, people from China, people from America, they always carry their own unique microbiome signatures. And these signatures remain with them even if they have spent a long time in a new country. In addition, stress, anxiety, depression, hypertension, these factors changes our lifestyle, they change our dietary lifestyle, and thereby directly or indirectly changing our microbiome. In addition, during early life, this is one of the foremost microbiome factors that influence our microbiome, including the C-section or preterm birth, the mode of birth, in addition to besides the mode of feeding during first 3 to 6 months of life. We have seen that children that are exclusively breastfed, they have a very different microbiome compared to children which are subjected to exposed to formula feed early in their life. So, and besides this, there are many other factors that may transiently or heavily influence the microbiome. And emerging studies now show clearly that whatever we have in the gut microbiome, it also crosstalk with our brain. And research has clearly shown that a healthy microbiome maintains a healthy intestinal epithelial integrity. And then there is a healthy crosstalk interface between gut and brains, and which is necessary for a healthy cerebral neurodegenerative health or central nervous system health. But studies are clearly showing now that if there is a dysbiosis in the gut because of diet, because of lifestyle, because of drugs or any other factor, this may lead to disintegrity of intestinal epithelial cell wall, which may lead to abnormal leakage of some inflammatory molecules, toxins, etc., or live viable bacteria, thereby inciting neuroinflammation and other problems in the brain, and thereby leading to an impaired neuropsychiatric health. So clearly, all these studies have shown that there is a two-way connection between aging and gut biome. While we know that gut biome, any dysbiosis in the gut biome increases the predisposition to a lot of diseases, not only of gut, like bowel disorders, colorectal cancer, and other problems, but also Parkinson's, Alzheimer's, hypertension, they have been associated with dysbiosis in the gut. But emerging study also shows that when a person is old, then aging-associated microbiome changes, such as reduced diversity and increased proliferation of opportunistic pathogens. These factors may also promote, deteriorate aging process. So they may increase the chances of infections and diseases during old age. So they're clearly showing that, yeah, there is a clear two-way connection between microbiome and aging. I have started recently doing some studies on mouse models of aging, and where we have clearly seen that even in mice, not only in human, even in mice, we have seen that old mice have very, very different microbiome compositions as compared to young mice. Even if they're from the same facility, they're maintaining the same facility, they maintain a normal diet, still old mice have a very different microbiome composition compared to young mice. We have seen that old mice have increased firmicutes, decreased bacteroidetes, and increased proteobacteria, which comprise a lot of opportunistic pathogens in the gut. So we have seen that even phylogenetically, these old mice have a very different composition of microbiome compared to young mice. At the same time, we have also seen that the response of a mouse gut microbiome to a low-fat or high-fat diet differs by host age. We have seen that young mice versus old mice, when they both are maintained on a low-fat diet, they have a very different response to this diet. At the same time, the young mice, their response, the microbiome response to high-fat diet is very different compared to the old mice when they are given a high-fat diet. And based on literature, many studies report increased firmicutes to bacteroidetes ratios as a indicator of obesity and type 2 diabetes. This increased firmicutes bacteria ratio has been associated with a lot of human and mouse studies on obesity and type 2 diabetes. And we found that this firmicute bacteria ratio was very, very high, especially in old mice when they were subjected to high-fat diet. So clearly showing that the dysbiosis of the gut microbiome because of high-fat diet is much more severe in old mice compared to young mice, probably because young mice have more resilience of the microbiome or more integrity of the microbiome. And then at the same time showing that old gut microbiome is more sensitive to this diet-induced gut dysbiosis. So to understand more about how different diets, and dietary patterns changes the microbiome, this was the first study that I did during my postdoctoral experience, where what we did was we had a subset of non-human primates, the African green monkeys, and we did a study wherein we maintained these monkeys on two different diets. One was on a Western style, typical Western style diet, and one group was maintained on a Mediterranean style diet. And these monkeys were maintained on these two diets for a very long time, like 1.5 years. So it was a very, very long and tightly controlled study. And we scanned their microbiome after this experiment. And what we found clearly was that the monkeys that were maintained on a Mediterranean style diet, they had a very, very different compositions of the microbiome compared to monkeys that were maintained on a Mediterranean diet. And clearly we found that the monkeys that were maintained on a Mediterranean diet, they had a significantly increased microbiome diversity. So their microbiome was more diverse compared to the other group of monkeys. So we performed a lot of machine learning algorithms to identify what is going at the level of bacterial genera and species. And we found a lot of bacteria that was significantly or uniquely influenced, regulated by the Mediterranean diet compared to some other bacteria that were uniquely modulated by Western style diet. And we finally found out that there are few selected bacteria. One of them is lactobacillus, which was significantly increased following the consumption of a Mediterranean style diet. And this lactobacillus has been considered as a very healthy member of our gut biome. And most of the probiotic strains available in the market, they actually belong to lactobacillus genus. So there was clearly an increased expansion of lactobacilli in the mouse gut in these monkeys following the Mediterranean diet. But in addition to lactobacillus, we found that few more bacteria were also significantly increased in these monkeys consuming the Mediterranean diet. And one of them is Faecalibacterium, which is an active fiber degrader in our gut and is an active butyrate producer in the gut. So it is also considered as a next generation probiotic bacteria. And there are a few companies now trying to isolate strains of Faecalibacterium and endorse them as probiotics. In addition, we found Oscillospira, Lachnospira, Dialystor, and a few more that was significantly increased. And all of these bacteria have been associated with a good, health, a good gut health in human studies as well. So clearly showing that how a long dietary pattern of Mediterranean diet flourished, the population of health beneficial bacteria, fiber degrading and butyrate producing bacteria in the gut of these monkeys, thereby boosting the overall diversity of the microbiome. We also had the data on the cardiovascular or cardiometabolic markers in these monkeys, for example, BMI, body weight, triglyceride, visceral fat, insulin resistance, cholesterol, and calories consumed. And when we correlated this data with the microbiome changes occurring in these monkeys, we found clearly and very interestingly that most of the bacteria, these green dots are the bacteria that were increased by Mediterranean diet. We found that most of the bacteria that were increased by Mediterranean diet, they were correlated negatively with most of these features of cardiovascular health, which means that the bacteria, the monkeys which has increased population of these bacteria because of Mediterranean diet, they had lower BMI, they have lower body weight, they have lower triglycerides and lower, insignificantly, but lower cholesterol and triglycerides. So clearly it was showing, this study showed that how diet-induced microbiome changes, are positively correlated with improvements in the cardiometabolic health markers in these monkeys. And in addition to cardiometabolic health markers, we also had the data on their like socioeconomic stress level markers in these monkeys, because these monkeys, they make their own hierarchical groups within their pans. So some of the monkeys are aggressive, some of the monkeys are subordinate or subdominant, and they receive a lot of depression, stress and aggression, which represents like a socioeconomic model of depression. So when we correlated the data of microbiome with these monkeys, again, we found that most of the bacteria that were fostered by Mediterranean diet, they were correlated with negatively with time spent alone, which is an indicator of loneliness in these monkeys. And these were negatively correlated with cortisol levels. At the same time, they were positively correlated with gait speed and grooming time, which is actually a healthy marker of socioeconomic status in these monkeys. So clearly showing that this Mediterranean diet boosted microbiome was correlated not only with improvement in the cardiovascular health, but also with neurological or socioeconomic depression and stress markers in these monkeys. So to replicate these findings in a clinical setting, we then did a pilot study in healthy human subjects, where we had a group of 20 subjects which were given an average American diet for a couple of weeks, followed by a switch to a Mediterranean style diet for another couple of weeks. And then we measured the microbiome. And we found that the microbiome diversity in following a couple of weeks on Mediterranean diet was clearly significantly increased. And so microbiome richness as well as species evenness in the gut of these people was increased within 2 weeks. So if you remember that we found the same pattern even in the monkeys that were consuming a Mediterranean diet. So clearly replicating that study and showing that yes, Mediterranean diet was able to increase the diversity in the gut of these humans within 2 weeks. In addition, we found that the composition, the deep composition of the microbiome was very, very different between the two groups. But we performed many more analyses to pinpoint, what are the main bacterial genera which are playing a role in this diet-induced microbiome changes. We finally came down to conclusion that Akkermansia was the one bacterium which was significantly increased following 2 weeks on Mediterranean diet. And this bacteria is like, again, a health beneficial bacteria. It has been, reported in many studies with healthy gut health. And it is a mucin degrading bacteria in our gut. So higher amount of this bacteria has been associated with improvement in, improved gut health and improved leakiness in the gut. And we found that Mediterranean diet increased the intestinal population of Akkermansia. In addition, it also improves the population of Lactococcus and Lachnospira in the gut of these people. In addition, there were few more bacteria which were not significantly increased, but their like Faecalibacterium was again increased in the gut of these people. And again, this is a bacterium which was increased also in case of non-human primates. So there were many signatures of the gut microbiome which were increased in the gut of these people and which were totally reproducible compared with our monkey studies. It's clearly showing that yes, this, first of all, it showed the success of that non-human primate model. I think that study is still going on, and we are now trying to do more, experiment on the same study. Like we are performing RNA-seq and fungal flora of those monkeys to identify at a deeper level what's going on at the metabolomic and transcriptomics level in the microbiome. So but together, our studies have shown that, for example, it has been long traditionally been known that Mediterranean diet, because of increased fiber levels, increased intake of short unsaturated fatty acid, and reduced processed foods, reduced saturated fats and calories, it has been associated with improvements in insulin sensitivity, immune functions, with obesity, type 2 diabetes, cardiovascular disease, etc. What our studies suggest that improvement in the microbiome diversity may also be one of the mechanisms by which this Mediterranean diet actually mediates these effects or impart these effects on gut and metabolic health. We have found that yes, Mediterranean diet improves the microbiome diversity, increases the population of several health beneficial bacteria while reducing the intestinal levels of several opportunistic pathogens. And we believe that this improvement in the gut biome may be one of the reasons by which this Mediterranean diet confers some of these benefits on host health. So one component of this Mediterranean style pattern is increased consumption of yogurt and probiotic food. So we were also interested in understanding deeper, like what's in the Mediterranean diet which changes the microbiome, which is governing these or regulating these effects on the microbiome. So in a study, what we did was, okay, let me just define it. Probiotics are live microorganisms which, when consumed in adequate amounts, confer health benefit to the host. And most of the genera belonging to these probiotics are Lactobacillus, Bifidobacteria, Lactococci, Leuconostoc, and Saccharomyces cerevisiae. And there are a lot of health benefits associated with a lot of bacterial probiotic strains available on the shelf. However, mechanisms of most of these probiotic strains are still not proven yet. And then whether and how these probiotics interact with the microbiome is not very clearly understood. So to address this gap, we did a small study wherein what we did was we actually isolated some of the probiotic strains from the gut of healthy babies. What we did, all these babies were born naturally and they were exclusively breastfed, and we selected these babies because we believe that this young baby's microbiome is nascent and purest form of microbiome associated with human health and unexposed to any environmental contaminants or diets or other things. But so we isolated like around 300 to 350 strains of bacteria, and based on a lot of battery of tests, we finally found 5 strains of probiotics which showed tremendous effect using in vitro models to be able to be considered probiotic. What we did was we put that microbiotic cocktail back into a mouse gut and we scanned the microbiome. And what we found that in control mice, there was no shift in the microbiome. But when we put these probiotics in the mouse gut, we found a significant shift in the mouse gut within 5 days to 2 weeks. We found that compared to 0 day, there was a change occurring in the microbiome composition within 5 days to 2 weeks and up to 3 to 5 weeks. In addition, we found that this probiotic feeding actually increased the bacterial diversity in the gut of these mice. In addition, we found increased levels of butyrate in the gut of these mice. So clearly showing that, and if you recall, the same patterns we were observed even in the monkeys and the pilot human study, we found the microbiome diversity was increased following Mediterranean diet also. And the butyrate producing bacteria were also increased. And here we directly measure the butyrate, and we found that this was increased in the gut of these mice following the probiotic cocktail. We did a bunch of more tests, and we found that the mice that were given this probiotic cocktail, they had improved glucose tolerance and improved reading of insulin tolerance. In addition, we found, a slightly reduced gut permeability in these mice, and showing an improvement in the leaky gut syndrome. And we found that in the intestinal tissues, in the colon tissues, we found reduced expression of interleukin-6 and interleukin-1 beta. We also did gene expression analysis of a bunch of other markers. We didn't find much difference, but in these two specific markers, we found significant reduction in the inflammatory genes. In addition, we found reduced crown-like structures in the adipose tissues of these mice, showing that there was a reduced inflammation in the adipose tissues. Plus, we performed a bunch of neurocognitive assays such as open cage test, Morris water maze test, and inclined screen test. And we found that mice that were given a probiotic cocktail, they always performed better on these tests compared to the mice that were given a placebo cocktail, showing that yes, probiotic cocktail was improved the microbiome diversity as well as improved the metabolic and neurocognitive health in these old mice. And we did a bunch of more studies where we did metabolomics and RNA-seq and combined this data with all the microbiome data also. And eventually we came down to conclusion that this probiotic cocktail was somehow reducing the microbiome dysbiosis by improving the bile salt hydrolase activity of the biome. We found that there was increased levels of conjugated bile acids in the gut of these mice. And there was one particular metabolite, which is taurine, was significantly high following the intake of this probiotic cocktail. And we did a lot of in vitro assays, and we verified that microbiome-derived taurine was actually the one metabolite which improved the tight expression of tight junction proteins in the mice as well as in the cell culture lines of intestinal cells. And we found that this taurine, the microbiome feeding as well as the taurine feeding, improved inflammatory markers in the intestinal tissues of these mice. And we believe that this is one of the pathways by which this probiotic cocktail was able to improve, promote healthy aging in terms of improved metabolic health and improved neurocognitive and physical function in this mice. We then wanted to take a better understanding of the molecular mechanisms, that why and how at bacterial level this is working. So what we found that probiotic feeding was increasing the population of one particular bacteria, which is Akkermansia. And Akkermansia is a mucin degrader in the gut. So we wanted to see what is it related to mucin in the intestine. So we measured the fecal mucin, and we found that yes, mice that were given the probiotics, they had increased levels of fecal mucin in their gut. And then we found that the expression of MUC2 mRNA gene, which relates to the mucin integrity in the intestine, we found that this expression of MUC2 gene was increased in most of the intestinal tissue, duodenum, jejunum, ileum, and colon. And then what we did was we heat killed the bacterium and then fractionated it into cell wall and cytoplasm to identify which factor of the bacteria is involved in this action. And we found that even in the heat kill form, the bacteria was able to increase the expression of MUC2. And then not the cytoplasm, but the cell wall component was the one that actually increased MUC2 expression. We then subfractionated this cell wall component into two further components, lipoic acid and peptidoglycan. And we found that lipoic acid was the one fraction that was actually showing that MUC2 expression effect in these mice. And then we found that this feeding of LTA alone in the mice reduced the expression of interleukin-6. And then we used a knockout model of toll-like receptor 2, and we found that this effect of LTA on interleukin-6 was abolished when we used the knockout model. So clearly we did bunch of more studies, and what we found was that this heat-killed lactobacillus paracasei strain, which was isolated in our lab, it was showing its effect because of its cell wall lipoic acid uh, moiety, which we can call like a postbiotic. So postbiotic is a new word, new term being used to refer to the metabolites or parts of the probiotics through which these probiotics work. And we found that one of the postbiotic may be cell wall lipoic acid, which works through TLR2 receptors, eventually leading to the increased expression of MUC2 gene, and thereby maintaining the homeostasis of mucin in the gut, which may be one of the reasons these mice had improved leakiness of the gut, improved inflammation, and eventually led to better, hallmarks of aging in different, as verified by different tests. Then another component which we are trying to focus right now is prebiotics. This is another important component of prebiotics. And prebiotics are indigestible ingredients that selectively, foster the growth of beneficial bacteria in the gut. And the most common prebiotics being researched are fiber, inulin, galacto-oligosaccharides, fructo-oligosaccharides, and resistant starches. And research, including ours, shows that the effect of these prebiotics on host health are mediated at least in part via modulating the microbiome. So one of our research is focused on understanding how different prebiotics actually shape the microbiome and affect our metabolic health. So we recently did a study where we had 4 groups of mice, wherein one mice was controlled, one mice was given inulin, the most popular and most widely studied prebiotics, and the other 2 groups received 2 novel, resistant starch and polysaccharides from plant sources, sago and acorn. So this study had 3 different types of prebiotics: fructo-oligosaccharide, galacto-oligosaccharides, and resistant starches. And what we found was all these 3 prebiotics led to a significant shift in the microbiome compared to control group. And all these 3 prebiotics had a different signatures, but all 3 increased the levels of bacteroidetes and lactobacilli, and a few more beneficial bacteria whose abundance was increased in these 3 groups of mice consuming 3 different prebiotics. And we also found that all the prebiotic, fed mice, they had improved glucose tolerance, significantly or insignificantly improved glucose tolerance. And then the gene expression of zonulin and occludin, the markers of a healthy gut permeability, was significantly increased following these prebiotics. In addition, we found that we performed a bunch of gene expression assays on the brain tissues from these mice, hypothalamic tissues, and we found that one particular gene, which is pro-opiomelanocortin, its expression was significantly increased by one particular resistant starch from corn, and from other 2 probiotics also, it was insignificantly but higher following the prebiotic intake. And we did a bunch of further studies to understand how it's working. And what we found was that these prebiotics, what they do is they reduces the pH of the fecal matter, and then by increasing the short chain fatty acids level in the gut. We found specifically propionate and butyrate levels were significantly higher in these mice. And these increased short chain fatty acids, including propionate and butyrate, they lead to improvement in gut permeability and reduces the leakiness by improving the expression of tight junction proteins in the intestinal epithelial wall, such as zonulin and occludin. And these leads to decreased systemic inflammation, thereby improving insulin sensitivity and reducing the markers of type 2 diabetes and obesity. However, we think that this might also be an indirect effect via regulation of nutrient signaling in the brain, because this pro-opiomelanocortin has been associated with nutrient signaling and food intake in the brain. Although in our study, we didn't find any difference in the diet intake in these different group of mice, but we still believe that there might be a direct effect of this gut microbiota boosted by prebiotics on the nutrient signaling in the brain, which may lead to improvement in insulin sensitivity and type 2 diabetes and obesity markers. In addition, we also did an in vitro microbiome analysis on these prebiotics, and we found that the effect of these prebiotics on the microbiome was very different in samples from young people versus samples from old people. So our studies clearly show that the effect of dietary fiber on the microbiome is very different between young versus old subjects. And then some of the signatures were very similar in the gut of in the samples from old people and young people, but there were some unique signatures such as Oscillosapyra, Coprococcus, and a few more bacteroids genera, which were increased only in old people and not in young people. At the same time, there were some signatures of the microbiome which were increased by the prebiotic, but only in young people and not in old people. So these in vitro and pilot study clearly showed that the effect of prebiotics or any dietary component on the microbiome might depend on host age and what is the native state of the microbiome. So considering the emerging importance of gut-brain axis, and a lot of studies are now showing that microbiome may be involved in the pathogenesis of Parkinson's, Alzheimer's, and a few more, and many other neurodegenerative diseases. So based on that literature, we recently did a study on Alzheimer patients, wherein what we did was we had a cohort of 17 patients with mild cognitive impairment. We wanted to see if there is a unique microbiome signature in these patients. So what we found was we found that the patients with mild cognitive impairment, they have lower microbiome richness, microbiome diversity in their gut compared to cognitively normal, participants, which were age matched. In addition, we found that the patients with cognitive impairment, they had significantly increased abundance of several bacteria, for example, Mogibacteria, Phoscolorchobacterium, and Proteobacteria, and Gamma Proteobacteria. And these were very interesting effects because these Proteobacteria and Gamma Proteobacteria are the families that comprise a lot of opportunistic pathogens within them. So it showed that the patients with cognitive impairment kind of had a more kind of proliferation of opportunistic pathogens in their gut. So which was like a negative signature of the microbiome healthiness in the gut. But what we found was that these differences in the microbiome, they were not restricted only to the microbiome of the bacteria. We found that these patients with cognitive impairment, they also had a very, very different fungal flora. There was few fungi that were significantly higher in the gut of these patients with cognitive impairment. And then we found that fungal diversity was also directly associated with several markers of Alzheimer's disease. We found that there were 4 to 5 fungal genera in the gut of these patients that were correlated significantly with the markers of Alzheimer's disease, such as amyloid beta 40 and 42 proteins, and total tau proteins, and phosphorylated tau proteins. And these markers were measured in the cerebrospinal fluid of these patients. And when we combined the data set, we found that there were few bacteria and fungi in the gut that were significantly correlated with brain markers of Alzheimer's disease. And we found that when integration of multiomics data of microbiome, bacterial microbiome, fungal microbiome, and metabolomics data clearly showed that compared to cognitively normal counterparts, the patients with cognitive impairment, they had a very, very different, networking and correlation or co-occurrence regulation between bacteria, fungi, and short chain fatty acids. So clearly showing that there is a dysbiosis of the whole gut microbial ecosystem in the gut of these patients. It is not just that the bacterial microbiome is different, but how bacteria, fungi, and metabolites, they connect to each other and regulate each other. This whole ecosystem was very different in the patients with cognitive impairment. Now, getting back to our focus on nutrition, in this same cohort, we then did dietary interventions. We chose to intervene with two different dietary patterns. One was a modified Mediterranean ketogenic diet, which is like a Mediterranean diet, but towards ketogenic formulation, which means that it has little lower carbohydrates and more unsaturated fatty acid intake. And we compare this diet with the effect of American Heart Association diet, which is a very low fat and high carb diet. So with this dietary intervention for 6 weeks in this cohort, and what we found was that American Heart Association diet didn't have any significant effect on the microbiome. But on the other hand, the Mediterranean ketogenic diet had broader effect on the microbiome in these subjects, with several bacteria getting affected significantly, like bifidobacterium, lactococcus, and lactococci were significantly modulated by the ketogenic Mediterranean diet. In addition, we found that this Mediterranean ketogenic diet increased the microbiome diversity during 6 weeks regimen, but this increase was seen only in patients with cognitive impairment. It was not seen in control subjects, because control subjects already had a high homeostatic or balanced microbiome diversity, whereas this MCI subjects had a declined or reduced diversity, which was kind of being recovered by these dietary interventions. In addition, we found this modified Mediterranean ketogenic diet increased the butyrate levels in these patients, but not the American Heart Association diet. So these two effects, increase in the microbiome diversity and butyrate level, are the same effect that we have seen also in the interventional studies using monkey models as well as mouse models. So again, replicating our all previous studies and showing that yes, how diet can improve the microbiome diversity and increase the abundance of beneficial short chain fatty acid in the gut. In addition, we found that all the bacterial sequences of bacterial communities associated directly with Alzheimer's disease was reduced following this dietary intervention with Mediterranean ketogenic diet. And not only with the Alzheimer's disease, but all the bacterial communities associated with type 1 and type 2 diabetes, those are also significantly reduced following the Mediterranean ketogenic diet, but not with the American Heart Association diet. In addition, we found that there was direct changes in the direct markers of Alzheimer's disease. We found significantly increased cerebrospinal fluid levels of amyloid beta 42, and we found insignificantly reduced cerebrospinal fluid levels of total tau proteins in these patients. And when we combine all this data, we found that there were several bacteria, Mogi bacteria, Rhinococcus, and Roseburia, which is again a butyrate producer. And in addition, there were some fungi such as Candida, Saccharomyces, and few more. They were significantly associated with changes occurring in the cerebrospinal fluid in these patients, in terms of the levels of the Alzheimer's biomarkers. So clearly showing how the diet modulates the microbiome in connection to the brain markers. So these studies clearly showed that, first of all, the patients with mild cognitive impairment, they had a very different microbiome compared to the controlled cognitively normal subjects. And the response of the microbiome to the dietary interventions was very different between a Mediterranean ketogenic diet versus the response to an American Heart Association diet. In addition, we found that this not American Heart Association diet, but the Mediterranean ketogenic diet beneficially modulated the microbiome and improved the Alzheimer's biomarkers in patients with cognitive impairment, clearly showing how diet, microbiome, or diet, gut, brain might be connected to each other. How what we eat not only changes our microbiome, but may also change our brain health. So overall, my research is focused on understanding how our gut is altered or senescent during old age, and how this gut dysbiosis leads to a leakiness of the gut metabolites or gut bacteria into the circulation, and how this during old age this increased dysbiosis and leakiness leads to the increased chances of increased susceptibility to brain problems due to neuroinflammation or direct or indirect effects due to decreased neurotransmitters or regulating hormones. And what we are trying to focus on is mainly the dietary intervention. Our focus is mainly on how high-fat diet and how sugar diets affect this gut-brain axis, and how we can improve this abnormal gut-brain axis using Mediterranean diet or hypertension, anti-hypertensive diet, or a combination of the two, like what we call it mind diet, how these dietary interventions fosters a healthy microbiome and metabolites in the gut, and how this healthy microbiome leads to improved integrity of the gastrointestinal epithelial integrity, and thereby leading to an improved homeostasis of the overall gut-brain connection. So right now, my laboratory at FSU is focusing on understanding the mechanisms of specific dietary elements on aging microbiome and intestinal health. And we are trying to understand how, different dietary fibers and starches, they impact the aging associated gut and cardiometabolic health, especially via microbiome. And then in addition, we are doing a study on using an Alzheimer's mouse model to study how this diet microbiome interactions might play a role in Alzheimer's pathology, and whether and how these dietary interventions may prevent or delay the onset of Alzheimer's symptoms. In addition, we are also doing some studies on understanding how maternal dietary and microbiome elements may, influence the infant microbiome dysbiosis and infants' predisposition to metabolic health. So I'm highly, thankful to all our funders, including ISL, including FSU CRC, which have given us some funds to do some pilot studies. In addition, like my studies are currently funded by USDA grants and a grant from the Yakult Probiotic company from Japan. So I would like to conclude my presentation here, and I'll be very happy to have questions and discussions from here on. Thank you very much.
Thank you. Appreciate it. We have lots of time for questions. So anybody want to jump in? Maybe stop sharing your screen for a little bit, and
yeah, sorry.
We can always reshare if we go back to your data.
I was targeting 50 minutes and
Well, you did a good job, actually. You got pretty close. Amazing. Okay.
I do have, this is Judy Delp. I do have a quick question, and you might have said it. Sorry, I got interrupted by a phone call for a little while I was listening. So your diets, are they isocaloric between the one you're comparing and the one, and then in terms of total, like, important critical nutrients, are they also matched? So then when you really compare one diet to another diet, I guess what I'm asking is, where is the big difference? Like, what are the things that are included? Is it just more fat? Is it more fat, more sugar, but everything else is relatively similar?
Yeah, so the major factors that were different were fiber and fatty acids. So for example, the Mediterranean pattern had more of unsaturated fatty acids, poly and monounsaturated fatty acids, whereas the Western style diet had a little more of saturated fat. The other big difference was the dietary fiber. The Mediterranean dietary pattern always had more of insoluble carbohydrates, inulin and other plant-based fibers, whereas the Western style diet has more of soluble fibers, soluble starch, and cellulose, etc. In addition, the Mediterranean dietary pattern had more of lean protein from fish and poultry and eggs, whereas in other we use lard and other natural sources.
And I'm always curious in general, when you do something like that and you do pair feeding or however you work it, how do you control how many calories one group is? Do they don't have the food ad libitum? Do you give it to them only once a day? Do you take it away once they've had a certain?
Yeah. So which particular study you're referring to? Because the way we feed, we provide this diet is very different based on monkey studies or human studies or mouse studies. like for the studies on non-human primates, that was my first study on Mediterranean diet. We controlled the calories and we also controlled the diet. We gave them food. They were trained monkeys actually, so we give them a plate, which is typical to what a human person eats. We give them one plate and they take like 30 minutes until they're done. Then we take back the plate. So we were ensuring that they consumed equal calories, and whatever we provided, they ate it all.
Okay.
Yeah, so the calories were controlled, the intake itself was controlled. So these monkeys were housed like in a big facility, but they had small feeding pans. So every time we need to provide them diet, we used to have a vet who was trained. He would just whistle, and this particular monkey will come, and then he'd sit in the pan, and he will eat quietly, nicely, and then when he's done, he will leave, and then so we were controlling for all those, dietary confounding factors. We were ensuring that whatever meal was prepared, it was close. Like first of all, these were designed with the help from our institutional dietetic people, and then whatever we were provided, we ensured that all these animals, they were consuming it right away. So we did as much as we could to control for diet or calorie intake in this.
And you always hear, and I'll let somebody else talk, I'm sorry, but you always hear from people who go on like change their diet. They always say, I feel like I have so much more energy, this, whatever. So any chance that any of these monkeys, for example, that you put on the diet are actually, I'm an exercise physiologist, so are they actually being more active? Do you have any way of evaluating their overall caloric expenditure?
So we didn't have any direct readout of any like physical activity, but what we observed was that there was a difference in their, clashes, hierarchical clashes. So monkeys that were given like a Mediterranean diet, somehow they were like, they had less clashes of their socioeconomic hierarchy, and there was less prevalence of aggression received by within the cohorts of these monkeys. So there were few, things that were improved in terms of their overall socio-economic status within their own pans. But we didn't have any direct marker of physical activity. But we found that the monkeys that were given a Mediterranean diet, they were moving more freely. They were less depressed because of their hierarchical, dominance and aggression, and they had reduced cortisol levels, they had reduced triglycerides, and they had less time spent alone, which is again an indicator of depression in these monkeys. So monkeys that were given Mediterranean diet, they were spending more time in the community, in the groups. So they were less stressed and more social.
Okay, thanks. Very nice, by the way.
Thank you.
Alice, we can't hear you. You're talking, but I don't hear a sound. Do you want to maybe just put the question in chat?
Unless that fixed it, I will do so.
It did. We can hear you.
That's exciting. That's
Okay.
a first. So Alice Pomodoro from Geriatrics at College of Medicine. This is very interesting. And I'm kind of assuming that the monkeys are a little bit younger and all cognitively intact for my question, because I have a dreadful time getting humans to eat what they're supposed to eat when they are cognitively intact age. And for those with mild cognitive impairment and early dementia, it gets even worse, especially if they are having their typical failure to thrive syndrome. So, for example, I had a lady came in who's been losing 10 pounds a year for the past 3 years, and her primary diet is Little Debbie snack cake right now. So when I envision trying to do a dietary intervention for some of these folks, I'm just having trouble with the translational aspect of it. They can make much better tasting fiber cookies than they do now.
Yeah, I totally get your point. Like it was harder for us also. For example, our studies on patients with cognitive impairment, it was like more challenging than doing any animal studies, of course. And I remember we had to involve local YMCA communities to help with, outreaching and, following up with the patients and ensuring that they had weekly meeting with the subjects and with ensuring the dietary adherence and that they're properly following the guidelines and the dietary things. And we were supplying them the olive oil and few other things from our own side, because we understand these when there is a old age, combined with dementia and cognitive impairment, it's very impractical to be, adherent to instructions on diets and nutrition. So it's always challenging. So we had a great team with YMCA, dietetics people, with clinicians to ensure that our data are like intact. Whatever we are proposing is being followed up.
So do you feel like they were adherent like maybe 2/3 of the time, which would still be fabulous if they were my patients, or that they ate only what you provided and not Little Debbie cakes too?
So we could not control for like each and everything they were eating in their free living conditions, like snacks and something, but we were giving them a daily chart of what should be the breakfast, what should be the lunch, what should be the dinner, how much oil to put, how much of what to eat. So, and we were following up every week or twice a week to make sure that they, with them or with their, guardians who to ensure that whatever we propose is being actually done and it's not getting affected by cognition or memory problems. Yeah.
Now, I was about to ask kind of a related question too. Do you know what the diets were? And Alice may have already answered that question. Do you know what the diets were before they entered your study? Because what you're trying to do is change the microbiota, and
So,
that would probably depend on what the current equilibrium was.
so you're talking about the human study that we did.
Yeah, the
right?
human studies.
Yeah. So yeah, they were like free living. We are not following up on what they were eating beforehand, but we had a control period of 6 weeks. So for that, we provided them control diet. All the people were on the same diet. So we wanted to have a washout period, of anything. So that during that 6 week, we ensure that it's stable. Whatever they're eating is stable in terms of calories and carb and fiber and fat and everything. So we had to have that washout period for sure, just to maintain that the microbiome is not confounded because of any prior variations in the microbiome.
Thanks. Other questions? We have 2 minutes left. That's it.
There's an observation in the chat, I think.
Oh, yeah.
And the same would be true for me with my Korean background about fermented vegetables.
Fermented vegetables are a big thing regarding diets.
Yeah, hi, this is the question from me. I'm a PhD student in psychology department. So do you have any opinion on this fermented food, or are you familiar with the research on that? In Eastern Europe, in Poland, it's very popular and everybody advises you to eat fermented food, yeah.
I don't have a particular experience with fermented vegetables in Poland, but yeah, we have the data from different ethnicities and different cultures, and every culture have their own ethnic, foods. Like South Asians, they consume a lot of kimchi and other pickles and fermented foods. Like if you go to Europe, there is increased consumption of kefir, yogurts, and their own versions of fermented products. In USA also, there are different versions. So I think fermented vegetables are a good source of fiber as well as probiotics, natural probiotics, because in my studies what we have seen that most of the probiotic strains, they don't work that effectively in clinical settings. They work great in animal models, in in vitro studies, but they have a high failure rate in clinical settings. But we have seen that fiber or prebiotics, their effects are more reproducible. Their effect on microbiome is consistent from one person to another person. Whereas when we consume probiotics alone, they may colonize the gut in some people, but they might not be able to colonize in other people. And some probiotic strain will work for you, but for some other person, that strain will not work, but some other strain might work. And so there is a very high individual specific variability in terms of probiotics alone. And that's the reason, like my research as well as other people are now endorsing symbiotic kind of approach, where focus more on diet and prebiotics things to naturally, flourish your indigenous probiotics. As long as we are healthy, we are doing fine, we are disease free, it means we are already carrying our own probiotics in the gut. So we don't really necessarily need to rely on commercial probiotics, because that's like a blindfolded thing. We don't know what is working or how it is working. But we have seen that when we control for our diet and lifestyle, when we maintain a healthy diet and healthy lifestyle, the effect on microbiome are consistent, long, and then more reproducible, and they fosters the natural colonization of natural population of indigenous probiotics. As we have seen in all my studies, whenever we give any cohort a Mediterranean style diet, there was an increase in expansion in the population of indigenous probiotics such as lactobacilli, bifidobacteria, fecalibacterium, and these effects were more prominent if we give only the probiotic cocktail. Clearly, so effect of diet was much more dominant than the probiotics alone. So in that way, ethnic foods, fermented vegetables, I think they're a good way to go, as long as there is less processing and all natural food.
Well, we're kind of at our deadline. We'll maybe, if Ravi can stay on a little bit longer, continue for a few more questions. But let me first thank you for a very wide-ranging and very informative presentation. Thank you so much.
Thank you, Dr. Jonathan, and thank you everyone for your time.
Yeah, that was excellent.
Thank you, Bill. Thank you, Kali.
Okay, if you can stand for just a minute or two, there might be maybe a few other quick questions, not sure.
Okay.
Almost everybody's gone. Okay,
Okay.
I'll let you go in a sec. I wanted to follow up on just one question I didn't have a chance to ask. So you had used knockout models suggesting that there are genetic components. Are there standard biomarker or genetic markers rather in humans that might indicate whether someone does a better job with diet A versus diet B? Because I would think, particularly if there isn't a lot of, and this is true for some populations, a lot of migration out of particular areas. People stay around in a particular area with a particular bacterial and fungal environment, and sort of from an evolutionary perspective, they may be adapted to that particular setting and diet. Although we've seen, I guess, with lactose intolerance that those things can be acquired or although I think the lactose intolerance literature has undergone some changes recently.
So whether it's lactose intolerance or gluten allergy, for example, they're also like very environmentally regulated. Like, for example, I come from India, and then we consume a lot of dairy and a lot of gluten. And even in the environment, like, we are immune to many of the things, but I bet when you go to India, to my hometown, you will definitely get sick. You won't be able to tolerate that food, that environmental or pollution, whatever factor. So yeah, there are many genetic factors also. Like in celiac diseases, in Alzheimer's, we have seen that sometimes a genetic defect may make us predisposed to a number of problems. But sometimes, even though we have a genetic predisposition, we have seen that ethnicities and like traditional dietary and environmental factors sometimes may help in decreasing the prevalence of those symptoms, or sometimes, and then now this new research has clearly shown is showing that like more and more defects coming because related to GLP pathways and interleukin-17 pathways. And some of these might also be associated with some other underlying genetic problems. So I think we need more studies, but genetics might play a role in predisposition or protection from a number of diseases.
Yeah, that's what I was wondering. Thank you. Okay, I won't hold you here any longer. Thanks again for an excellent presentation, and we'll stay in touch.
Thank you. My pleasure.
Have a good holiday.
Happy holidays to you all. Thank you.
Bye-bye.